So, but we better let the meeting start, hadn't we?
Yep, we can get started. So first of all, I'd like to express my deep appreciation to all of you that found your way to Zoom tonight. I really enjoy working with everyone at Mycological Society. And I'll give it to you, Mike, to introduce our guest.
Well, we've been talking to Rod here, Rod Toulos, Amanita researcher. There's not much more to say. I think those of you who heard Rod talk in the beginning know, I don't know of anyone who doesn't know Rod. I'm not really prepared to do this. I thought Teresa would, but Rod has been researching Amanitas since, you said, the late 70s. Why don't I turn it over to you, if you could just give us 2 minutes before you start by telling us a little bit about your interest in sequencing and how that happened.
Okay. I don't want to scare you all away, although I'm going to talk about sequencing a little bit in the talk, and I do rely on sequencing very much to deal with something as difficult as the section vaginatae, the grisettes of section of vaginatae of Amanita. What happened was that in the late 1990s, when GenBank numbers began with an A, you can imagine that now, we're up to MZ, I think. At that time, I think they were AF. At any rate, I was asked by a person who I had taught English to, and for whom I was a foreign reader of their PhD thesis, namely Dr. Yang from Kunming. He asked me if I would send him a bunch of American species because they were going to extract DNA and build a phylogenetic tree. I barely understood. And so I had my sequences, some of my sequences in GenBank begin with AF. But at that time, I was just, feeding them specimens. In 2012, the very beginning of the year, there was a meeting chaired by Dr. Tom Bruns from Berkeley to discuss a amateur-based project to increase the number of DNA sequences of fungi. And that turned into the Mycoflora project, which now has another name. At any rate, I was invited to, can you do what?
No, I think that was done on purpose.
Oh.
Yeah.
I'm seeing what everybody else is seeing. Jeez, what an ugly looking guy. Pandemic hair.
Yeah. But we love your poinsettias.
Hair. Anyway, at this meeting in 2012, I was told that my website was sui generis, and that's what Tom wanted everybody to have for all the genera. And to make that happen, he thought making all the amateurs, or making it possible for all the amateurs to get DNA sequences would encourage people to create these websites that would be like a monograph online that's always changing, which is in fact my idea of what I was doing. And so I thought, he likes my website, I better get it to fungi too. So slowly, I got involved with sequencing. Dr. Karen Hughes wanted to have a project where we dealt with rubescent taxa in North America and Europe. And so one project after another came along. The study of the transition from non-mycorrhizal to mycorrhizal was a study where they wanted amanitas, and so I got involved in that. And finally, I got to the point where I could derive the sequences from the raw data myself. And in fact, I prefer that to do that now. So that's where it all came from. And now we have something like 3,000 sequences listed available through the WOW website. And they are for species that aren't even named yet. But we're really trying to cover, at least cover North America real well, from coast to coast. And we'll do that until I can't do it anymore. That's essentially the game. So what I'd like to do, unless somebody has, very important thing. Teresa is watching the chat. If you find I've gone too fast, there's something you want to know more about, and it's possible for me to cover it in a brief time before delivering the rest of the talk, I'd like to know, because you may not be the only person feeling that way. And I've never delivered this talk before too. I just wrote it for this, for tonight. So if anything is not, you can't understand, send a chat message to Teresa. She'll gather it up together and she'll give me a signal or tell me that, break in and tell me that there are questions that need to be answered right away. And that's what we'll do. Okay. All right. Let's see what can happen here. I'm ready to share.
Says host disabled.
Yeah, the host has disabled the screen share. Nope.
Pascal, could you set up Rod as a co-host? So he can share?
Yes.
There we go. Where am I? Is this it?
Yeah. That's it.
Okay, let's go back to the top of the thing. And let's do the slideshow. Okay. Here's our show for tonight. I'm going to talk just about the section Vaginati because it's a hard section. And it was just ignored for the better part of a couple centuries, except for an occasional European author. And there are no type specimens for a lot of species, and we're just starting to get around to fixing problems like that. But I want to talk just about the west coast of North America, species in the southern Canadian provinces, well, even some in Alaska, but yeah, let's say Alaska to Baja. And so there are not that many species that have been named and worked up. And so we can do it in an evening. And I propose to do it in the following way. First, to provide exactly what we mean by the Vaginati. There are 7 sections to the genus Amanita. And in terms of the views that I learned from Dr. Boss, there's other people now that are dividing up more sections, but I want to stick with the basics here. For years, I've taught the 7 sections at amateur societies because knowing the section allows you to communicate with each other. You don't know necessarily the species. In fact, in North America, it's very likely that you pick up a species and it doesn't have a name. And so what you need to be able to do is say, I have something brown here. It's got a vulva at the base of the stem. The vulva does not deteriorate rapidly. It stays white, but it gets orange dots on it. The cap color is brown, and there's a white band around the cap or a cream colored band around the cap. And I don't know what it is, but I do know it's in section vaginatae. And you can begin to have a conversation or post something on Mushroom Observer by just using a section name. I flatter myself that I had something to do with that. The next part of the talk is just to give you a feeling of how the numbers, the count of species has changed just in the last 60 years. Then we're going to take a look at some ways of grouping of taxa so that we can talk about what taxa the Western North American taxa are like, what group of other taxa they belong in. And finally, I'll show you one way of categorizing some of the Amanitas in section vaginatae by using just information that would be in a basic barcode. It's not part of the ITS gene, but it very frequently shows up just after it always shows up just after the barcode gene ends. And I discovered the fact that in the section vaginatae, there are 2 groups that can be distinguished by that little pattern of 15 or so letters that occur immediately after the barcode gene ends. Now, then I want to talk about things organized according to a very rough single gene tree. I don't want to call it a phylogeny. We do not know enough about these species, nor do we have enough genes of them to do anything that is sophisticated as a phylogeny. What we have is a pseudo-phylogeny created by a piece of software that thinks it's creating a phylogeny, but has inadequate numbers of species, and we don't have any idea how many it ought to have. So you'll see in a minute that I've created a very cartoony phylogenetic tree to use in the talk. Finally, I just want to mention the work of Tsui. The C is a TS sound, published in 2018, who has subdivided everything much farther. And I'm not going to use his work tonight, even though there's a lot of interesting stuff in it. It's just too much. It goes beyond what I've taught online and to folks like members of the Mycological Society of San Francisco. So to characterize the vaginatae, we can use some basic things. First thing is, if you say, well, it's a member of the Amanitaceae, it's a member of the family Amanitaceae, what does it have to to be a member of that family? Well, you have to have a stem that has tissue in it that looks like here, where I move my cursor. This is called a longitudinally acrophysolytic tissue. The acrophysolyds are these club-like cells, and they are aligned vertically in the stem, and they're terminal on the ends of branches of hyphae, like you see the wall here at the end of the hyphae. These are acrophysolyds. Acrophysolyds are really important to Amanita because Amanita starts as a button and inflates itself by putting water into inflatable cells like the acrophysolyds. The same cells cause the cap to expand. The same kind of cells cause the stem to elongate and to broaden. Now, in the Amanitaceae, we're in talking about something in the genus Amanita. And the unique thing that makes an Amanita, it has nothing to do with whether the gills are free from the stem. There are Amanitas with gills that touch the stem, that are attached to the stem. All that stuff is stuff that's made up for field guides. It gets passed on and changed from one author to the next, and pretty soon it's a lie. I've seen field guides that say all gills of Amanitas are white. It's not true. You saw an orange gilled Amanita on the first slide. Anyway, all Amanitas do have one thing that no other agaric has, and that is it have schizohymeneal ontogeny. Schizohymeneal ontogeny means they start out as a button of completely uniform tissue. And unlike all other agaricales, they don't make a stem first, then a cap, and then gills that grow down into empty space from the cap. In this button, the first thing that happens is a vulva forms around it. Then the cap skin comes, and if the cap skin is going to get a color, it gets a color very early on. Then you get little shadows that suggest where the cap is going to be and where the stem is going to be. And then later than that, you get shadows of where the gills are going to be. And the gills form as a solid donut around the top of the stem. They're not split apart. That's what schizohymeneal means. Schizohymeneal means that the surface that's going to be spore bearing has to be made from a solid donut, and it has to be split into gills. Schizo is splitting. The hymenium is the surface that bears the gills, the spores. So schizohymeneal ontogeny means development. So this schizohymeneal ontogeny means that there's a vulva, there's a sterile gill, the gill margin is sterile, and there are no basidia on the gill edge because it used to be attached to the stem. And it had to break away by using single-use Velcro, and that Velcro gets left on the stem, which are lines at the top of the stem, and left on the edge of the cap, which is that so-called fimbriate material on the edge of the stem. And finally, there can't be any cystidia because cystidia, by definition, grow on an exterior surface. There is no exterior surface on an Amanita until it's after it's finished expanding, but it's not making more cells at that time. It doesn't grow cystidia. So it can't have cystidia. Finally, the section Vaginata is one of 7 sections of the genus Amanita. And it is in the subgenus that has spores that do not turn color with Melcher's reagent, in amyloid spores. So we come down here, we get to here, we satisfy these conditions. We also have in amyloid spores. Now, there are two more characters. One is that the stem is totally elongating. There is no bulb at the base. And the Caesarii, mushrooms like Vernacocera, also have totally elongating stems, but you need one more character to get rid of them. And that is that at the base of a basidium, the cell that makes the spores, there's a little cell on its side in the Caesarii. There's a little cell on the side that connects it to the cell just below it, a little extra cell. And that's called a clamp. And in the Vaginata, you don't have them. I've never seen one. They might be there, but I've never seen one. Okay, so now we know what the Vaginata are, very roughly speaking. Now, counting the number of Amanita. In 1961, the Dictionary of Fungi said there were estimated 200 species of Amanita all told. When I bought that in 1978, they said, what? Because already people had published books that had 125 new species in them. So that had gotten out of date pretty fast. So in the year 2000, Boss and Dr. Boss, my mentor, and I were invited to give a paper to the Bolletino Gruppo Micologico Bresadola. And we said there, we thought that we probably reached 1,000 species of section vaginata. And at that time, we estimated there were 600 that were either described or known provisionally at the species rank or below. And now we can say the following. On the WOW site right now, the www.amanitaceae.org site, we have 1,103 amanitas, 612 have been published and accepted, 152 are provisional, and 290 have temporary codes that I gave them. Now, other people may give things temporary codes, but they don't tell me about it, so I don't know about those. And I probably couldn't keep track of it anyway. The vaginatae alone contain 407 taxa at or below species rank, 134 published and accepted, 95 provisional, 168 temporary codes, and rapidly growing. So we're looking at a group here that was minimally understood 60 years ago, and now has grown. So there's 407 taxa that have been published in it. So that's quite a change. Now I'm going to start talking about grouping. And we're going to be strictly talking about vaginatae now. Now, in this picture, in the upper right-hand corner is a picture drawn by Connie Borodenko when she is astonished to find this mushroom, of which a good 9-10 inches was underground. This is about the part that was above ground, above that point. There was immense vulva on it. It had an umbo in the center of the cap, like the thing that Trempetomyces species used to crash out of termite colonies. Essentially, they break through concrete with an umbo like this. It's called a perforatorium. I never saw a perforatorium on an amanita before. I said, my God, this is just an amazing thing. And that weekend, we collected white-capped ones, brown-capped ones, vergate cap, that means looks like hair coming out from the center. All of it turned out to be one species, Amanita penetratrix, the penetrating amanita. It comes from way, way underground and blasts its way through roots and everything else to get out. And when we got DNA for Amanita penetratrix, we discovered that nobody had ever told anyone about the way that the barcode gene ended and the large subunit, which is the immediately subsequent gene, began. Everyone told me it begins TTGACC, blah, blah, blah. It turns out that in the vaginatae, you have two choices. You can begin TCT or you can begin TTTD. And these can be separated from the Caesarii because they don't have the middle letter. And so this was something that just accidentally came up because I was very interested in looking at these things called the motif. Oh, I see a misspelling. That should be motif, not motfi. But, okay, anyway, the motif just means a recognizable pattern in the DNA sequence. And motifs are very often used to recognize the beginning or end of a gene, especially the ribosomal genes, as opposed to protein coding genes. Anyway, that's not important to understanding what's going on here. The important thing here is there's a little trick that I, a novice, happened to notice. And so Amanita penetratrix this special, the special motif is present in 30% of the vaginati that are known today and that have been sequenced. And the other 70% are this kind. Now, on the West Coast, you don't have penetratrix, but you do have this guy who has a penetratrix-like sequence. And I called it Castelli because one of the first collections came from Castella in California. Its range, and today I just found out that it occurs in Oregon. So its range extends from Oregon to Arizona. It's often called a pale Pachycolia, because I think maybe because of the dark ring around the inner end of the striations on the cap. But it's quite distinct from all other known Amanitas. Pachycolia is a problem for me because I've never been able to get a sequence out of any collection that I have. So it's my picture of Pachycolia, which is not great, it comes at the end of the talk because I don't know how to categorize it. Now, there's another, and here's another one where I don't have a picture, but I think I know what it looks like. At a Soma Foray, we had a magnificent collection of Constricta, but on examination, one of the specimens in the collection was not Constricta. It looked like Constricta in a lot of ways. It had a dark brown cap. It was rather large. This was a collection of very large Constrictas. And the one thing that might differentiate it would be that the vulva at the base of the stem was not turning gray. I have only a single collection of it, but we do have DNA. So, and it has this TCT pattern in the DNA. So I want to put it out here as another species on the Western list. Now, for a couple of weeks, I've been trying, well, actually for longer than that, for months, I've been working with a fellow in France on trees made from only the barcode gene and only constrained to be the vaginati. And we have made enormous trees. And every time you add another species to it, especially from North America, the likelihood you're going to add something that's not like anything in the tree, and the whole tree adjusts, tree building software adjusts the tree. And so it's very, very hard to say what the tree looks like. I got so frustrated trying to deal with it, making a tree that I could put in this talk, I decided I'll just make a cartoon of a tree. These are the positions that groups that I've named fall into in that tree, and these are their relationships. And if I make the tree again tomorrow and put 3 more species in it, it won't look this way. But I want to point out that the green indicates that the TCT motif is present in that section. The first 2 sections, these represent what you might call the earliest diverging, which some people associate with the oldest the oldest ancestor for the tree, and Penetratrix and another species called Insinuans, which has ranges from about Maryland to the Gulf Coast. They both have the TCT, and Insinuans has a ring on the stem. Now, the next diverging thing here you see is Amida hausmannii, and that's European. And it's a very small group, and nothing on the West Coast falls in that group. The red diamond is going to mean members of that, there's 1 or more members of the group that has a representative on the western coast of the United States, Canada, or Mexico, and that I know about. I try to know as much as I can, but it's hard to keep up. So we're going to go through these groups. I'm just going to call them groups. This group contains the genus Amanita umbrino-lutea. This group contains Amanita longicuneus, which is a provisional name. This contains Amanita constricta and has lots and lots of gray weak volvas in this. Amanita alceades is a recently described species from France. It's a very small group, but you have a member of that group on the West Coast. Villosa is alone in a group. So the idea that it might be related to Pseudocrocia or another orangish member of the Vaginata is apparently not true. Crassiputamen is named, the name means a thick nutshell, and it has very thick shelled inflated cells. And it is, Crassiputamen itself was found in California. It was an unidentified specimen in a bunch of stuff I borrowed from the Thiers Herbarium. So I described it in great detail, but didn't give it a name because I only knew of one collection. We've been able to get DNA from that original collection, and we now find this thing everywhere. And apparently it's another one of these things with a very variable cap color, from very, very pale to yellowish cream to deep brown. So then there's nothing in Lividopalescens or Dryophila or Supravolvata groups. But when you get to Bruneo Philogenea, we have at least one member of that group in California. I mean, on the West Coast. Origin seems to me that has a range from Oregon and Washington south. Mayeri is a European species, and it has a very small group that we know of. Corylli is, or coralie, is named for the tree Corylus, which it is associated with, although it also grows under birches. And we have several species related to Corylus in North America, and some of them are on the West Coast. Corylus has the TCT motif. Simulans is a recently redefined, well, actually not redefined, a new neotype had to be created for it, but now it's turning up all over Europe. And we have a number of Simulans-like species in North America, including some on the West Coast, and about half of the Simulans group is a subgroup that contains the TCT motif. The Phreobilis, which is European, but you'll recognize maybe a species from Pacific Northwest and British Columbia that's been called Phreobilis and might be Phreobilis if only we knew what Phreobilis was. And then there's a group that I gave the name to, this Pseudocrocia, which is an eastern species, but there's at least one very curious Pseudocrocia group species on the West Coast. And then this is an northeastern species that is not represented out here. So that's the idea of what this is about. This is an outline of the next bunch of slides, which are going to be pictures of fungi. In the constrictor group, is that right? Is that the first one? Yeah, it is. In the constrictor group, there are three taxa on the West Coast: Callisaurora, constricta, and Constrictosaurora, which will be on the next slide. Callisaurora, and this is a Debbie Weiss photograph, I'm pretty sure, is like Cecilii in that it has a friable, this is not an annulus, this is part of the vulva that got left at the around here and got moved up the cap by expansion of the, moved up the stem by expansion of the stem. You can see the vulva is graying and weakly structured, which is like the European Caeciliae. This was one of the things called constricta in California until we realized that it didn't look that much like it. And here's a constricta cap over here for contrast. This is the vulva of a constricta, and this is the black here is the shadow of this vulval limb being cast by the sunlight, which is over this direction. Here's another constricta, and you can see that over time, the vulva becomes gray. And that is typical of almost everything in the constricta group, and I'm sorry, almost everything in the Caeciliae group, and consequently in the constricta group. You can see that the top of the vulva flares here, and you can see that the vulva was pushed away from clinging close to the stem by the expansion of the cap. Even here, you can see the bulge right there. The cap expansion must have really taken effect when the base of the cap was that high from the bottom. And you see it started to push the vulva, bulge it out. And the same thing has happened here. The top of the vulva has been bulged out. So this is two of the constricta group species. And here's the third, Amanita constricta sorora. And I'm not going to try to remember who all the photographers were, but I think this might be Ron. No, I don't think so. But I give credit to all the photographers at the end. I want to, because without the people who use Mushroom Observer, and without people who correspond to me, without people who dry specimens and send them to me, I wouldn't be able to do what I do. And I consider the fact that they do it a second time pay. I mean, I don't get any money for doing any of this, and I never have. But my pay is because people will come back to me a second time and say, would you look at this one? So constricta sorora, you can see it also has a vulva that's weakly structured, but it leaves more of a patch on the cap. It's a lighter brown and at least sometimes has a white margin. And it looks like here, it maybe it's paler when it's young. So this is Amanita constricta sorora, and it's in California. Here's some more. Now this is from the Umbrino lutea group. Now on your right-hand side over here, you have species NW04. I want to acknowledge the fact that a lot of people call this species here Pachycolia. And I know that because people have sent it to people in China who got their material from North America have this, and they called it Pachycolia. So my image of Pachycolia is not like this, but it could be I'm wrong. So there's an unknown situation there. I'd love to have some material of Pachycolia, robust, big, dark Pachycolia. So I'm sure what I've got. Now this species is called C29, and it's a very robust. I mean, this looks like a russula with a vulva. I mean, it has pink gills too. And this also was discovered relatively recently on Mushroom Observer. This character here over here has dark edges of the gills. This is the bottom of the cap, and these NW04 or perhaps Pachycolia is these black edges. Now the Alciades group, there are two. One of them is Oregon 3 and the other is California 25. And I want to call your attention to in California 25, which has kind of fluffy pieces of vulva all over the cap. The stem ends right there, you can see the end of the stem. The two sides of the stem are curved at their bottoms, and then the vulva is below that. In this case, the bottom of the vulva is conical. That is really unusual. I've seen it 2 or 3 times. And they're not all in the same section. So this kind of obconic, that means a cone upside down, obconic vulva, clearly separated from the bottom of the stems, is evolved multiple times. This OR0, we've never seen the bottom of it, OR03. But one thing we can tell, its cap is different from this. It has a membranous vulva, which is very tight to a pale gray cap. That's not an annual stem cracking. Okay. Here's Amanita villosa. This is also, I remember, is a Debbie V sequence. Picture. And you all know that. It's, as I said, it's in a group all by itself. The Bruneo fuliginea group, Bruneo fuliginea was described from China, and many other species described from South Asia and Europe have the same exact barcode. To the extent that one European author has combined all of the species into this name. Species that I was planning to name for Amanita Lindgren, Janet Lindgren, it falls into this group very convincingly. Now here's Crassiputamen. You see, this is a young one. This is in this, if we had this large enough on your screen, you could see that the inflated cells here have double walls. They have a thick wall, and that's the thick nutshell. Also, as we were discussing before the lecture, it has a pretty robust vulva and has left a large calyptra or patch on the cap. The smaller patches have been left on the older material, and you can see that it appears to get more brown with age. That's Amanita crassiputamen. For a long time, I didn't know what it looked like until Mushroom Observer was invented. Then I saw it. Now the Corylli or group includes Amanita species constricta 01, which I don't have an image for. But again, this is the same one we mentioned before, that it must look like constricta because it was in a mistaken for constricta and collected with constricta. So if you can see, unfortunately, people's pictures are on top of this part of my slide, but the text here is satisfactory description of the situation. And Amanita species C31 is given over here. I don't know what the mature cap color is, but it looks like pigment is developing there. I only know it from this one collection. Now here's Amanita chiricahua. It was first found in Chiricahua County, Arizona. And I did this over here because these are from 35 millimeter slides. And this did not blow up well. And so I said, I'll leave it at the small size and let it go bigger behind it in order to fill space. But it's a very interesting species. And it also has the TCT motif. Now, curiously enough, in the Simulans group, we have things with the TCT motif, like Castelli, which I showed you previously, and Amanita protecta, which does not have a TCT motif. This is the holotype of Amanita protecta collected in Southern California. This also is from a recent collection from Southern California, and it's more mature probably than this, or maybe in a moisture zone. And the cap is not gray. It's another case of another species in California that has a white variant. The DNA checks out the two genes I use. Beautiful matches. Now here's from the Friabilis group. This Friabilis is a European taxon. In Europe, the Norwegians say it's one species and the French say it's another. And so it was originally described from Scandinavia. So I would hope that we could find another case from Finland or Sweden or Norway, so that we can get a better idea of what it is. This is very like the species that's called Friabilis in France. At first I thought it was different, but over the years I'm leaning more and more to think that it may. This one is from the northwestern states of the US, and this is one from British Columbia. This thing is in the Friabilis group. Amazing, crazy thing. It looks like a bag of rags exploded. And this blue color is from the fact that it was a very shiny cap and it was open sky, and the blue of the sky is reflected on it. And this one is another one collected at the same time, and you can see it's grayer, but there is some blue tint in it also. But this is really a remarkable species. Unbelievable. Now, in the Pseudocrocia group, there is something in Oregon, at least, that is in the Pseudocrocia group. This is where you would think that something like Villosa would end up, but no, it doesn't. And this is a dark center and an orange cap, and that glare there it was covered with goo. It was very viscid. The next one is in the Pseudocrocia group. This is from California, and it has a grayish or grayish brown cap, but a big white patch that covers most of the cap apparently. And you look down in the bottom right-hand corner here, and you see the stem tissue coming to an end above a pale orange conical vulva. Remember that? We had it before. And even the tissue in the middle of the stem is staining slightly orange here. We know from one photographed collection, and we may be getting some more other collections of it. But it's quite remarkable. Of the two species on the West Coast, they're not very closely related at all. And there's a third species that we know from Missouri, which has has a conical bulb that's as sharp as a pencil. And it looks like a sharpened pencil below the stem. And it doesn't seem to be related to either of them. So there must be some evolutionary pressure that Vaginatae can respond to by creating a conical vulva, but it's not clear what that is, obviously. Okay, not assigned to a section, Pachycolia. This specimen was given to me in an amazingly large mayonnaise jar by John Lenny, who had been keeping it in his refrigerator, saving it until I could get to California. This is my concept. Very dark cap with a dark ring, even darker ring at the top of the striations. Bulbs staining orange, rusty colored. This was really overdid it because it was in a jar in the refrigerator and got moisture in there. I think that's what contributed to this. a very dark stem. And I'd love to get at something else that looked like this and see if it was this or species NW04, in which case I have to give up NW04 and call it Pachycolia. But there's one of the puzzles that's left. Now, at this point, we can see there are at least 20, maybe 21 or 22 species of Agenata in the West Coast states and provinces. And the phylogenetic groups represented are each have a few species in them, but there are a lot of different groups with these species in them. So it's a high degree of apparent endemic novelty with very few, that word is holarctic, I think, I can't even see it, but that means common around the world in the Northern Hemisphere. And the Western species seem to be mostly strictly Western, with a very few exceptions, and very few North American and Western. I would say there's an uncommon degree of novelty, partially because of the lack of study of the group. And many taxa, finally, many taxa have pigmentless caps or pigment or pigmentless color variants of other taxa. The number of white caps is rather large. So we clearly have a long way to go in nailing, getting a good grip on the Vaginate. Let me see who these criminals were. Yeah, Callus aurora was Debbie. Where's Ron? Crassibutamen, C31, C32, and W04. Thanks, Ron. Cindy Trupovitz from Southern California is down here. I don't know this person very well, but he's the person that produced Oregano 3, and that was a fascinating collection. Christian Schwartz is pitching in here. Brittany Ramsey from Oregon was that the orange-capped species with the sticky top. A couple of photographs of collections came from Sava. So I want to thank all of these people and the people who sent me dried specimens who did not, whose pictures are not used here. It's been a terrific experience working with material. I've tremendously enjoyed the opportunities I had to collect in California. I don't know whether that's all over for me, but these people have been so kind and generous with me. I really want them to know how much I appreciate what they've done. And I thank you all very much. And end with this, it's something to meditate over, a large circular picture. The Amanitas are up here. This is the history of all the agarics, starting with the jelly fungi back in the Carboniferous. And these are the ages of the Earth, geological periods. And the agarics begin here, and they started from 182 to 156 million years ago. And you can see Amanita here, and there's the Amanitaceae beginning there, and that's Limacella, I assume. And so somewhere in here, mycorrhizal fungi were reinvented by Amanita. But there must have been a really dry period in here sometime in the early Quaternary that scared the Amanitas to death, and they just don't want to come up. They have to have a lot of rain and they wait for it. Anyway, thank you very much for your attention. And I'm glad to see that some people followed it.
There were no comments. So if you have questions, now is the time to raise them. You can unmute yourself and ask your question.
Is that chart that you just showed available somewhere? That's highly interesting.
Rod, is all your research from North America? Do you do any stuff in Europe or overseas at all?
No, I have worked with people in lots of countries overseas. We published species from India and Pakistan. I've co-authored papers about the genetics of poisonous amanitas with authors in China. I've supplied a lot of material to China, which sometimes gets a co-authorship. I've worked with people in Japan, most of European countries. My website now covers a lot of European amanitas, because when the Facebook page Amanita of Europe started up, they invited me in early on, and the same thing happened in North America. Facebook page for Amanita North America. So I've been able to use both of those as teaching vehicles. And when I see something fascinating, I say, "Did you dry it?" and if they say yes, I did, I said, "Do you have more than one photograph? Would you like me to look at the DNA of it?" And a lot of people are very interested in that, and it's quite expensive. It takes a lot of euros to send a little brown package with a mushroom in it to the United States, and I'm astonished that people do that. I work with people in South Africa. They have lots of cool amycorrhizal amanitas. I've gotten really into that. So, and I think I have at least temporarily defeated the idea that the genus should be split. After I'm gone, they'll probably do it again. But anyway,
yeah.
But right now, we're going through a phase where a lot of the work in developing countries, or countries where science is developing, they're beginning to have access to DNA sequencing, but their understanding of the process and the fact that there's a market for people who want to know the mushrooms, not the sequences. Sequences are fun, but what's really interesting is to explain to people how to know it when they see it, without DNA sequencing. And I think that is still a major challenge. And I explained to one of my colleagues in China, I said, "You're developing a middle class. What happens with the middle class is they become bird watchers. They become mushroom collectors. They try to learn what their grandmother knew." And so there's a market for information about fungi that's presented on a macromorphological level. And that's a real customer base. And for anything you do, there'll be many more people trying to find your fungus in the forest than there are going to be identified by gene, or to continue your research. Incidentally, much of this research doesn't get continued. It's done one time and then they pass on to something else. And so people don't go back and change the names on sequences in GenBank. And there's a lot of garbage in there. I mean, they have a sequence, yeah, but it wasn't magic. If you couldn't recognize the fungus, you put it on the wrong fungus. So at one point, 60% of the Amanita virosa in GenBank was not Amanita virosa. that's largely because it was from the United States and we don't have it here. So it's a funny thing. Science is a very funny thing. It's driven by grants in universities, which means that once you've done something, you don't do it a second time. Who's going to pay you to do it a second time? So everything gets left in a half-baked state. I was an engineer before I was this. In fact, I got a degree in mathematics at Berkeley. And it was called Logic, and was one of these back in the day of multi-subject matter degrees. It was Logic and the Methodology of Science. Anyway, it prepared me to become an engineer. And I became an electronic engineer for 25 years at Bell Laboratories when I couldn't get a job in a university. And so then I started a nonprofit and preserved farmland in New Jersey. And all during the time, from the time I got promoted in Bell Laboratories, I did mushrooms. So I did mushrooms, like I said at the beginning, since the end of the 70s. And I actually started trying to identify them about the mid-70s. Very frustrated because the stuff I found was never in the book.
Rod, we have a question about Vaginotic group. Are any of these species toxic?
Not in the section Vaginati. In fact, in section Vaginati and Caesarii, in most countries in the world, the representative species of those sections in those countries are eaten. I know of one case where it's reported that children get diarrhea from eating a member of the Caesarii, but the recipe that's being used, there's only one thing to eat at this particular, in the monsoon season. These people, the people that are in the group that is involved with this mushroom, normally eat bats and fruit. And when it's the monsoon season, there are no flowers, the bats aren't flying, the bats won't fly into their nets. So what they have on hand is dried fruit and this one mushroom that they trust. And they're called the Chepang people. And I named the mushroom Amanita Chepangiana. Or no, Chepangii. Wait a minute, it's not Chepangiana. Yeah, Chepangiana. Yeah, I was afraid I was saying Chepangensis, but no. Somehow my old memories of old mistakes come back late at night. Anyway, Chepangiana, the recipe they eat it with is peppers, lots of hot peppers, and dried fruit. Now, I think that would give kids diarrhea right there. So I'm not sure it's the mushroom causing that problem. But you do hear about people eating things that look like they're in the Vaginati and getting poisoned. But that happens in very remote places, and I don't know who is the person who is the giving the information, and I don't know what the mushroom looks like. So it's very questionable information. I don't in the world of European colonies and ex-European colonies like Mexico, Canada, United States, where mushrooms are eaten, they follow the same rules that are followed in Europe, and they don't seem to be problems. I think all the grisettes that can be eaten, and many of them have been.
I have a question about, well, somebody is asking about your phylogenetic tree here of all the species. They're wondering if that's published and available for meditation.
This circular picture here,
yeah,
it's in the paper that was in the biographical note that was sent out about me.
Okay.
This is from the paper that's mentioned in there. It was a study. It was trying to render the history of all agarics from as far back as you could go, with really long sequences of the large subunit. Now, they wouldn't accept a sequence less than 900 characters long. And so they were using, they used the same set of primers that the folks working with me use. And so I ended up, they originally said, give us 13 Amanitas, and said, you've got to be kidding. And eventually the thing evolved and evolved and evolved, and it was going to be the 3,000 agarics project. Well, then it became the 5,000 agarics project. Then it went back to the jelly fungi. So it was an immense project. And my role in it was to supply them a large number of specimens, discuss the sampling process with them, and work out a methodology to get good coverage for Amanita, and then try to fill in gaps, and then go over their DNA with them and try to see if they, because sometimes they had people, not me, had collected specimens for the project, and the DNA did not match the name. So I went through all of that with them, sequence by sequence, that whole blue part there. And very enlightening, very, very interesting project. And they were kind enough to put my name somewhere on the list of authors, somewhere down at the bottom.
I have another question. I'm wondering what you would recommend that we do to improve the quality of the material that's held in GenBank, and whether or not there's a way that we could clean up some of the existing material there.
There's a real hooker on that. I mean, there's a real caveat on that. The only person that can change the name on a gene is the person that submitted that gene.
The sequence. And
if they're too busy, it doesn't happen. If they're too busy writing the next grant, it sure isn't going to happen. Luckily, I don't have any worry about a next grant. And so I actually, every time I find a better name for something, I fix it in GenBank. And you can tell because the GenBank number, when it's first put in, the number has a 0.1 after it.
Uh-huh.
And if somebody changes it, it gets a 0.2. You can see my changes by looking at the 0.2s. It's not that much, but we prevented a lot of things, bad things happening up in here when we went over it before it got published.
That's good. It's really hard to try to look at in-group and out-group genetically on a tree if you don't have good quality sequences.
No, you're not going to make much progress, because one of the things you do if you're doing a project like this is you trim all the sequences down so that you have the core part. You might, for example, in the middle of the barcode gene, there's a subunit called 5.8S, and 5.8S is almost invariant. If you have 100 Amanitas, you'll have lots of change. You make an alignment. Change is happening on the left-hand side. Change is happening on the right-hand side. In the middle, very little change is happening because that's 5.8S. And so some people cut 5.8S out so that things will run faster, and so there'll be less obscure reasons for changes to occur. And when I was making the trees that are behind my cartoon tree, I was using the whole ITS gene. And I used 302 sequences to make that tree. And it was, I mean, the problem is that in some species, the gene varies a little bit. And in some multiple gene species have the same gene. That's the problem with ITS.
Mm-hmm.
I mean, it was one of the reasons why people fought over what the barcode gene would be for a decade or two. And then somebody just said, it's going to be ITS, damn it.
Mm-hmm.
And that's what we got. And LSU sort of is the backup, you know, the secondary. And even if with the two of them, you still get cases where things that really don't look like the same thing turn different colors in the refrigerator. I mean, have all different kinds of properties. And the ITS genes are the same, and the LSU genes are so close together. It's very difficult to tell. There's the crassiputamen, the thick nutshell Amanita. That has an LSU that's almost exactly the same as 5 or 6 other species. I call it the shared gene. They hardly differ at all. And you ordinarily would say 0.1% change. I'm not going to, 0.1% difference, I'm not going to worry about that. Or 0.2%. Some of these things that change, when you're looking at 2,000 characters, having two characters change is, what is that, 0.1%.
Yeah.
And,
Is there another region in the genome that you like to look at to help split these apart?
Pardon me?
Is there another region in the genome?
Yes. If I had the time, and time is really my constraining factor. If I had the time, and if I had a colleague who wanted to spend the time on it, a wonderful paper was written with another paper written with my material by a fellow named Santiago Sanchez Ramirez, who was part of his PhD thesis. He took a year from the PhD, stopped his PhD work, and looked just at the Amanita caesarii and searching for a protein coding gene that was variable enough to separate species, but not so variable as to create chaos.
Uh-huh.
And he used mathematical techniques to evaluate his thing, and he found 2 such genes. And we had done a paper together on the caesarii that used, let me see if I remember correctly, 4 protein genes and the large subunit. And we got results that just couldn't be right. He said, Rod, just tell me what can't be right. You know the species, you know where they come from. I don't know where they are on Earth's geologically. So tell me stuff that you think oughtn't to be together, that's put together, and that's put together and oughtn't to be together. And so I went over the stuff with him, and he said, good, I'm going to take a year off, and I'm going to find 2 more protein coding genes. We're going to throw LSU out. And he did a second paper, and darned if the paper doesn't place a species from northwestern Mexico all together in northwestern Mexico, connects them up to the species in California that look just like them, and they had been separated completely in the previous paper. And the problem with the large subunit gene, it produces things called long branches in a tree. And a long branch means it has no close relatives. And having no close relatives means that part of the DNA sequence could look like something far away. And it could look like something else far away in another direction. What happens is a black hole effect. Stuff from all over what should be spread out of the tree all gets sucked in around the long branch. And LSU is terrible that way. And I have told people this again and again, and there are people that still base the core of their research on LSU and a few protein coding genes. I keep saying, why don't you do what Santiago did? He got a beautiful tree that put geography and DNA intuitively together, all together beautifully. And that was a real test. And you guys are just doing what you learned how to do in grad school over and over and over again with no change. That burns me up. I mean, I'm an engineer. I only expect to get 85%. But golly, these guys are blowing it. So
could I ask a question on that? Sorry, it was another lifetime ago, but I studied systematic botany at the New York Botanical Garden, and the cynic in me when you said that you know, a new protein coding gene had matched your expectation, it was like, well, okay, so you found what you were looking for, but really, my guess is that it was a little more rigorous than that, and that you were just looking at more traits, right? So in addition to the genetic traits, you were looking at the morphological traits and the biogeographical traits. Is that a fair assessment?
Oh, yes. I mean, it was a phylogeographic paper. It was, how did American Caesaria get where they are? Where did they come from? And what is their, and so it really was about geology, geography. And so the genes were selected for their property of being able to differentiate taxa. They were specific to the Caesarii. He only looked in members of the Caesarii for these genes. And what he was looking for was enough variation so that you can build a tree, but not so much variation as you have chaos. LSU is chaotic. ITS is rather chaotic. In fact, in the year 2000, Tom Bruns published a little paper saying, you should not be using this gene that I invented, or this gene that I found for making phylogenies. It won't work. And strange that nobody seems to have paid attention. Well, some people pay attention, and some people in lesser schools don't pay attention. In fact, don't even read the literature. That's the problem. That's another thing. It's another thing we're losing. We're losing attention to the mycological literature. People don't know the literature back more than mid 1990s. There's 250 years of mycological literature. Finding the gems is a problem, but they are out there, and that's how you go. That's the way you communicate with people. You have to go back to the level that a person has learned, which is usually morphological, and then come forward as far as they want to go. But it's just a shame to not have the literature and to remove things like X when somebody has validated somebody else's name. The current trend in nomenclature is to throw out all of the stuff that they now think is pure history, only history. I'm sorry, nomenclature is 100% about history. It's about who named something first. And if you throw away the information that where the name was created and who validated it and when and who it's just a pity that only want the living people's names on things. It's ridiculous.
We have one more question about samples.
Yes.
Wendy is asking if you would like some dried Ammonita samples with pictures from Zambia.
I have had some in the past, and I would love to see what comes up. And now I have worked with a Zambian material that was from David Aurora, and I've worked with and also in those days you could get into Zimbabwe too, that material is really fascinating, and I really want to ask people that want to send me dried material that it be in the section vaginata. I really can't go on doing the whole genus. I'll do what I can. If things come up and there's some kind of an issue, I get tempted easily and I go over and work on something else. But I really want to try to do the vaginata. I have material from Nigeria, coal black ammonitas. It's just astonishing. Vaginata, coal black. And I've already, on my website, I have a provisionally named Nigerian species called Ife-Ifeensis, because it was collected at a university in Ife. But yes, delighted. The transparency, in the interest of transparency, with the pandemic going, it's really hard to get samples made and sent off to the sequencing shops. And I got one shipment back in December, and I've got most of the sequences derived, but some are still going. But if I could get another shipment out, I would send it out. Always, I would send another shipment out. The worst thing that can happen is that I get run over by a garbage truck with the sequence out. Somebody else will work on it. I want to keep going.
There are a lot of amateurs learning to do fungal barcoding.
Yes.
And I meet more and more of them.
Yes, I have a publication that I want to get out. It's already been circulated. I taught a course from it in North Carolina, and I also taught a course from it at the New York Mycological Society. And what it does is tells you how to do all the steps in deriving a sequence from the raw data, and also how to tell what you really have. In other words, it also teaches the motifs. So you know whether you have a whole barcode or a fragment and which fragment you have. So people in the New York Mycological Society really like that. And we did a 6-hour day of a seminar on a Saturday, and ran through a shortened version of that training session. And I did a 3-day version in North Carolina. I think the short one did the trick pretty well. But in other words, I have material on the Ammonite of North America Facebook page. There is a copy of a document that I used in teaching that in the files folder.
I'd like to find that. Would you have any interest in running a workshop like that out here on the West Coast, or maybe running it by Zoom?
I'm willing to try it by Zoom. I'm not going to be traveling.
Nobody's traveling right now.
Yeah, well, actually, I know people who travel, but I'm diabetic and I have a load of the so-called comorbidities, and I am not taking any chances.
Yeah, no, we wouldn't want you to. So when you did that training, I hope people aren't, she says, thank you, I'll send you only the Veganata group. What was I going to say? I was going to say, I forgot.
Oh, was it the gene derivation, deriving genes training thing?
Yes.
I have a set of slides, actually. I'll have to go look. But the New York thing was presented by slides, and we even had ultra electron microscope photographs of genes, and including the sequence of genes from which in Amanitas anyway, there are about 30 copies of the barcode gene on every copy of the gene. There are 30 copies of the barcode, and so this is one of the reasons why you get chaotic ITS sequences, because they may not all agree. Mutations can actually occur of one of the copies and not the others. And so what happens is, if you get Sanger sequencing, it's like trying to record a chorus, and all the copies of the ITS are singing, but they have different sheets of music sometimes. And when that happens, you get ITS sequences that have great gaps in them.
Yeah. Because
for that period, they're out of sync with each other, and the sequences are not the same, and it's producing junk. And so a picture that shows the series of genes, a sequence of the, what do they call it? The repeat of the ITS sequence is enlightening. It makes you realize that you're taking 30 copies out of a nest, something that's more like Medusa's hair, and those 30 copies for the species you're working on just may not all be the same. And then you get something I call the two mesas phenomena. The gene starts out right, looking pretty good, high quality from the forward read, and it starts out pretty good on the reverse read. Both collapse, and you have two blue mesas of quality with a desert in between with a few cacti. And you're really lucky when you get two blue mesas that overlap, and then you can get a whole sequence out of it.
I'm kind of hoping that as more people take an interest in fungal genomics, that we can find some more stable, conserved areas that we can work with. Mm-hmm. Besides what you've talked about. That's what everybody works with now, pretty much. And like you said, it's not that effective. And I think Tom, I think he first published some of those gene areas. It's been like 20 years ago.
Oh, he published them more than 20 years ago. 20 years ago, he said, uh-oh, it doesn't work the way you think it does.
Yeah, so there must be improvements since then.
Well, oh, yeah, I mean, I think the improvement you see, most people working with genes nowadays, they always try to go to the most expensive machine. So you're doing things like whole genomes from fossil men and women, humans or pre-humans. And that's very fashionable. And you can get a lot of money for doing that. And, but mushrooms is not got that same kind of draw.
Yeah, we noticed.
Santiago Sanchez's father is a mammals zoologist, and he works with antelopes in Baja California. And Santiago quickly went over and did antelopes and rattlesnakes. And as soon as he had finished working on the Caesarii, he stopped working on fungi to a great degree. And he went to Germany and worked at the place there that does all the prehistoric, well, actually more than prehistoric, early hominids. So he's had great training now. I don't know what he's going to do.
Well, I think there's a lot of budding mycologists that are learning to do sequencing, and they're learning to analyze specific regions. So I'm looking forward to innovations from these new young mycologists. There's a lot of them, and some of them are amateurs.
Yeah. Yes. I'm an amateur. I've never been paid to do mycology.
Well, we're so glad that you do.
Okay. Well, it's a lot of fun, and I have enjoyed this evening. You must have practically no audience at this point.
No, we still have 18.
Oh my gosh.
Yeah, we still have 18, and you know, it's late for late for Rod there. He's 3 hours ahead of us. So if you have more questions, let's get them on the table now, and then we can let him go off to bed. Thank you. It was a great Zoom. Okay. Anybody else? Very interesting. Fabulous.
Somebody's exaggerating there.
No, I don't think they are. It was really informative. I really enjoyed it, and I've learned quite a bit. So I'm hoping that I'm speaking for the whole group.
Well, I really wanted people to see all these strange, curious new species, and that was the first thing that I wanted to accomplish. And you pointed out to me at the beginning that I need to explain sections, and so I put that in as you suggested.
Well, I think it clarified things for me a great deal.
Okay.
All right.
I really enjoyed it. Thank you very much for inviting me.
We're happy to have you.
Thank you very much, Rod.
I don't recognize all the voices, but thank you very much for staying with it.